The cardiovascular system is an extremely important system from several standpoints, and one of the main things being that it is what truly connects all of our body, because we have to send blood almost everywhere in the body because it's delivering all the contents that all the cells at every level needs. So what is the way that we get the blood to go to all these different parts of our body? And of course, probably the most well-known part of the cardiovascular system would be the heart itself. So looking here at our heart, we see that the heart is about a fist-sized organ. It has a top area called a base, and the base is, where the major blood vessels are running in or out of the heart. And the bottom portion, or the more pointed area, is called the apex. And then we know the heart has 4 chambers in it, 2 on the right side and 2 on the left side. Now we're going to talk about those chambers and about the different parts. That's actually more of a lab subject, but we will be mentioning those. But right now what we want to also look at is the fact that we have our membrane around the heart called the pericardium. And we can see that part of the pericardium actually adheres to the heart itself. So that portion of the pericardium is called the visceral pericardium, and that's like an onion skin thin layer adhering to the heart muscle. Then we have the thicker portion of the pericardium, which is the parietal pericardium, and then we have a fibrous very outer portion that is actually on the parietal pericardium. So if we cut the sac off the heart, we normally cut off this outer area, and normally this visceral area is still adhering to the heart itself. Okay, so the heart is protected by this sac that's around it, pericardium. We actually do have like in most serous membranes, we do have this space between the outer parietal area and the inner area adhering to the heart, the visceral area. Okay, looking at the heart, we notice that we have 4 chambers. We have our right atrium, our left atrium, our right ventricle, and our left ventricle. We also have major blood vessels running to the heart or taking blood away from the heart. So anything that takes blood to the heart is a vein, such as the vena cava. Notice this is taking blood to the heart. to the heart, and to the heart, here and here, and it would also come in on this side. These are veins. Anything that takes blood away from the heart are arteries. So this is an artery called the pulmonary artery because it's taking blood from the heart to the pulmonary area, which is the lungs. And this is the aorta, which is taking blood away from the heart. So these areas that are taking blood away from the heart are arteries. These areas that are taking blood to the heart are veins. Okay, now, one thing we have to remember is that we actually have what we call systemic arteries and pulmonary arteries. We have systemic veins and pulmonary veins. Systemic means it's taking blood to most of the body's systems. Pulmonary means it's taking blood to or from the lungs. So the real definition of arteries is they take blood away. This is a pulmonary artery because it's taking blood away from the heart to the lungs. But the blood in here happens to be oxygen poor. The reason we're going to the lungs is to pick up oxygen. The aorta is the largest artery in the body, but it is a systemic artery, and all the other systemic arteries branch off of it, either directly or indirectly. So this systemic artery is full of oxygen, and that's what we typically think of arteries. Now here we see a systemic vein, the vena cava, and systemic veins tend to actually be oxygen poor. But this vein, and veins are by definition being blood carried to the heart. So I have a systemic vein, but here notice I have a pulmonary vein because it's bringing blood back from the lungs where it picked up oxygen, so that blood's going to be oxygen rich. And so pulmonary veins are going to bring it back from both sides actually, even though we only see it in the picture from this side, to the left atrium, and that blood would be oxygen rich, even though it's actually a vein, but in this case it's a pulmonary vein. Okay, now, so the pathway of blood through the heart, which is important, and of course blood is entering the right side of the heart and the left side of the heart at the same time, and blood is being squeezed from the right side of the heart through the pulmonary veins, pulmonary arteries, and blood is being squeezed through the aorta at the same time. And blood is coming back through the pulmonary veins and the vena cava at the same time. So, but let's just trace the pathway of blood. A drop of blood, let's say, that starts at the superior vena cava. So blood from the superior vena cava would enter into the right atrium. It would pass through the first of our 4 valves, the atrioventricular tricuspid valve, into the right ventricle. Then it would pass through the pulmonary semilunar valve into what's called the pulmonary trunk through the pulmonary arteries. Blood would then circulate to both lungs. It would then come back via the pulmonary veins, and we can follow these arrows here to see that. We would also have them coming back from our right lung over here in this direction and entering into the left atrium from the other side. And so we'd have oxygenated blood coming back from the lungs, passing through the mitral valve, also called the atrioventricular bicuspid valve, down into the most muscular portion of the heart, the left ventricle. Then blood would actually go up through another semilunar valve, which we kind of see part of a cusp right here. It looks like the pulmonary semilunar valve, but it's called the aortic semilunar valve because it sits at the base of the aorta. All right, blood would pass through the valve, go into the aorta, and be distributed to the rest of the body. Now, what allows all of this to happen is something called the intrinsic conducting system. Here they just call it the conducting system. This conducting system is made up of autorhythmic or nodal cells that form either nodes or pathways. So notice that we have what we call our natural pacemaker, which is a sinoatrial or SA node, which sits up in the right atrium near the entrance of the superior vena cava. This is actually where heartbeat really starts. This is where the signal that coordinates and begins each heartbeat begins. All right, the signal starts in the cells in here by a process called spontaneous depolarization, which we will talk about in a couple of minutes. But the signal is initiated here. It runs down through these pathways called internodal pathways, which also run across through the interatrial septum that separates this atrium from that atrium. So we have some of the signal running across here, and the signal then runs down the internodal pathways to the next node called the AV node or atrioventricular node. Now this picture makes it look kind of like the AV node is sitting maybe in the ventricles, but in reality it's sitting in the wall of the right atrium still, and then this area called the AV bundle is actually the first place where the signal crosses from the atrium into the ventricles. Okay, now the signal again starts at the SA node, goes through the internodal pathways, goes to the AV node. Then there's actually an important delay in the signal that occurs at the AV node. And after that delay, extremely short but important delay, the signal goes into the AV bundle, where it then runs down the interventricular septum and through the pathway of these areas called bundle branches, which notice I have a bundle branch that basically runs down the septum towards the right side of the heart. I have a bundle branch that runs down the septum towards the left side of the heart. Notice also have some offshoots off of that signal that basically, in this case, it runs through something called a moderator band to the base of some muscles called papillary muscles. Now, as the signal runs down towards the apex of the heart here, then it fans back up into the myocardial muscle areas of both the right ventricle and left ventricle, this again being the most muscular portion of the heart, and the signal runs down and then runs back up. Now, Why are we doing this? This is kind of like the heart's own nervous system. Even though we do have neural connections from outside, this is an internal system that initiates the signals, and they are electrical signals. But as the signal moves from cell to cell to cell to cell through the nodes and the pathways, it's also moving across into cardiac muscle cells. The purpose of that is if I move the signal to a muscle cell, it causes a contraction. Then it moves from cell to cell to cell to cell, each cell having an extremely short period of contraction that sends the signal to the next cell through the intercalated discs that control the cells, through little proteins called gap junctions that control those cells. Now, the purpose of that is, the heart's purpose is to actually pump blood from the heart to either to the lungs or to the rest of the body, and then to receive blood back from those two places. So what I need to do is be able to contract all these muscles down here and down here at the same time, and contract these muscles here and here at the same time. I can only do that if I coordinate that contraction. Well, I coordinate the contraction by starting a signal that runs from here down and down through here, but at the same time runs across to cells that are connected to the cells in the intrinsic conducting system. So it doesn't do me any good if I have a contraction here and a contraction here and a contraction here and a contraction here randomly. What I need to do is I first need to squeeze the blood from the atria to the ventricles, and then squeeze the blood from the ventricles out. So I need to coordinate the contraction of all of these muscles here and all these muscles here to squeeze blood out and out. I need to coordinate the muscles here and here to squeeze blood back down in there so I can squeeze the blood out. Now, so this is the system that does that, because the muscles will not basically contract just by themselves. They need a signal running to them. So these are the cells that spontaneously depolarize or spontaneously start and then send the signal through this pathway. Now, if we're to look at what the signal graph, the trace of the signal looks like, notice here are our millivolts. We can measure inside the cells, these autorhythmic or nodal cells, and notice that we have a resting membrane potential of about negative 60 millivolts. We have a threshold of negative 40 millivolts, and then here we see our zero, so here we see our positive numbers begin after zero. Now, what this means is that in this graph line right here, which is the same thing starting over right here, is actually called our prepotential, or this is where our spontaneous depolarization occurs. What that actually means is that in these cells, we have sodium leak channels. They're always open, so sodium is always slowly leaking in, and we can see as this grows, we can see that sodium is leaking into the cells at that point. Well, when sodium leaks in, because those sodium leak channels are always open, once we leak in and we take our millivolts from negative 60 to negative 40, because sodium's a positive ion, so as more comes in, it's going to build our voltage towards a more positive direction. Once we build that voltage up to threshold, then threshold actually opens up voltage-gated channels, which allow calcium to rush in. So this portion, once I reach threshold, threshold's the magic number, so to speak, that is the voltage that opens up the voltage-gated calcium. Calcium rushes in at that point, and we actually cross zero, which means we reverse polarity. We go from a negative to a positive once we cross zero. Well, notice it doesn't do that too much. Everything's always trying to reach equilibrium. So calcium coming in is trying to reach equilibrium on both sides of the membrane, just like sodium coming in was trying to reach equilibrium on both sides of the membrane. But we never reach equilibrium, but what happens instead is now we actually reach a point where our potassium channels open up, and so this segment where calcium's coming in is called depolarization. We're going in a very positive direction quickly. When potassium channels open up, we know there's always more potassium inside of cells than outside, so potassium starts leaving, and that is our repolarization. Now, at some point after this happens a couple of times, we also have our sodium-potassium pumps, which You know, all that sodium that came in, we got to pump it out, and all that potassium that left, we got to pump it back in. We also even have calcium pumps. So the calcium that came in, we pump it